Dear viewers, greetings. In this video, we are going to see about the cattle infecting bacteria Streptococcus agalactiae. First, general characteristics. Streptococcus agalactiae is a gram-positive spherical shaped bacteria and it is arranged in the form of chains and usually in pairs, and it is non-motile in nature and its size ranging from 0.5 micrometer to 2 micrometer in diameter, and it belongs to the family Streptococcaceae, capsules absent and endospores absent. And regarding the Landsfield classification, and the Streptococcus agalactiae belongs to the group B. So it is also called as group B Streptococcus. The mode of respiration is aerobic or facultative anaerobic mode of respiration. Optimum temperature for the Streptococcus agalactiae cultivation is 37 degrees Celsius and optimum pH is 7. And normally this Streptococcus agalactiae are present in the upper respiratory tract and genitourinary tract of the human beings, and this Streptococcus agalactiae was discovered by the scientist and American microbiologist Rebecca Lansfield in the year 1930 from the milk and cow with bovine mastitis. Disease transmission of Streptococcus agalactiae. Streptococcus agalactiae infections are mainly transmitted by two modes. The first mode of transmission is person to person spread by respiratory droplets, and the second mode of transmission is through the breaks in the skin after direct contact with the infected person's fomites or arthropod vectors. Virulence factors of Streptococcus agalactiae. The ability of the bacteria to cause the disease is called as virulence, and the term virulence was first coined by the scientist Louis Pasteur. And virulence factors are the factors which are responsible for causing the infection or diseases in the human being. And the Staphylococcus agalactiae produce two virulence factors and infects the human beings. And the two virulence factors of the Streptococcus agalactiae are capsular polysaccharides, which is rich in sialic acid, and the toxin beta hemolysin. The incubation period of the Streptococcus agalactiae is 7 days, and the infective dose required to cause infection in the human beings is variable. Pathogenesis of Streptococcus agalactiae. Streptococcus agalactiae originally discovered as a cause of bovine mastitis in the cattle, and it is a part of the normal bacterial flora colonizing the gastrointestinal tract and genitourinary tract of the significant proportion of the human population. However, it occasionally becomes an infectious pathogen colonizing the uterus, blood, brain and meninges. Like Streptococcus pyogenes, Streptococcus agalactiae has multiple mechanisms for avoiding opsonization and the ciliating process, phagocytosis. Streptococcus agalactiae pathogen is one of the leading cause of invasive infections in non-pregnant immunocompromised individuals and also causes bacteremia, septicemia, meningitis and pneumonia. Next, clinical diseases of Streptococcus agalactiae. Streptococcus agalactiae are responsible for causing four types of clinical infection. The first clinical infection is early onset neonatal disease. The second clinical disease is late onset neonatal disease. And the third clinical disease is infection in pregnant women. And the fourth clinical disease is infection in other adult patients. The first clinical disease caused by the Streptococcus agalactiae is early onset neonatal disease. Early onset neonatal disease occurs within 7 days of birth, and in this case infected newborn babies develops signs and symptoms of pneumonia, meningitis and sepsis. The second clinical disease caused by the Streptococcus agalactiae is late onset neonatal disease, and this disease occurs more than 1 week after birth, and in this disease condition the neonates develops the signs and symptoms of bacteremia with meningitis. The third clinical disease caused by the Streptococcus agalactiae is infections in pregnant women. In this case, most often the symptoms like postpartum endometritis or the pregnancy infection, wound infections, urinary tract infections, bacteremia, and disseminated complications may occur. And finally, in the adult patients, the Streptococcus agalactiae shows localized skin infection with pain, inflammation, lymph node enlargement, and systemic symptoms. Lab diagnosis of Streptococcus agalactiae. The first diagnostic method is microscopic examination. Under gram staining, Streptococcus agalactiae shows violet colored gram positive cocci and which was arranged in the form of chains. And in hanging drop method, the Streptococcus agalactiae cells are non-motile in nature. And colony morphology in culture medium. In blood agar, Streptococcus agalactiae was observed as grayish white, transparent to translucent, matte or glossy, smooth, flat, large and zone of beta hemolysis. In McConkey agar, the growth of the Streptococcus agalactiae is variable. And this image shows the beta hemolysis of Streptococcus agalactiae in the blood agar. And next, biochemical test. Catalase test negative, oxidase test negative, urease test negative, indole test negative, methyl red test or MR test negative, Voges-Prescott test or VP test positive, citrate utilization test negative, starch hydrolysis test negative, casein hydrolysis test negative, alkaline phosphatase activity positive, arginine decarboxylase positive, hyaluronase test positive, CAMP test positive, hepatic hydrolysis test positive, basitracin test resistant and POR test negative. POR test is the major test used for the differentiation of Streptococcus pyogenes from Streptococcus agalactiae because the Streptococcus pyogenes shows positive reaction for the POR test. And another test is CAMP test is also the important test for differentiating the Streptococcus pyogenes from Streptococcus agalactiae. For CAMP test, Streptococcus agalactiae shows positive reaction, but the Streptococcus pyogenes shows negative reaction. Finally, treatment and prevention. Penicillin G is the drug of choice. The combination of penicillin and aminoglycoside is used in patients with serious infections. Cephalosporins or vancomycin is used for the patients who are allergic to the penicillin. And for high-risk babies, penicillin is given at least 4 hours before the delivery. And finally, no vaccine is currently available for the control of Streptococcus agalactiae infection. Dear viewers, that's all about the bacterium Streptococcus agalactiae. Thank you for your support. Thank you.